I'm here with Isuru Silva, who is studying in Dr. Barton's lab and is a good friend of mine and possibly one of the smartest people I've ever met.
You're too kind.
Yeah. And you actually did research in undergrad.
Yeah, so I had 3 papers back in Sri Lanka. No, 3, 7 papers.
7 papers.
7 research papers. And I think I'm the one who had at least 1 paper during my undergrad time among all my colleagues.
Wow, that's a lot for undergrad.
Yeah, I had 7 papers. 3 of them are directly related to microbiology, and others are just random stuff like elephants, birds, dragonflies, because I love nature.
Well prepared for the PhD, I guess, because you've done all that already.
Just try to, I tried my best to practice myself, to write, publish, because that's what we need in the PhD, other than for lab stuff.
That's awesome. So now that you're in graduate school and doing the PhD, what do you study specifically now?
Yeah, so for now, it's again astrobiology project, and then it's in the cave science field, and then also into microbiology. The thing is, in astrobiology, it's not just one niche. There are so many subfields in the subject of astrobiology. So now I can just say it's a cave microbiology project addressing one of the problems in astrobiology. So to be exact, I would say I'm trying to understand the highest limit of wavelengths, or the longest wavelength which can support phototrophic life for the energy requirement. So if I'm trying to simplify my words, I would say, so we can categorize light based on the wavelength. So let's say visible light, infrared light, like that way. So for now, what we know is 720, 740 is the highest wavelength that we know so far for general photosynthesis. That's the highest wavelength that life can harvest for the energy requirements. So I'm trying to see, is there any other wavelength that can support for the phototrophic life? So for that, my model environment is caves. Because in caves, because of the refraction and reflection of wavelengths, because of the cave wall properties, they reflect more near-infrared light compared to visible light region. So as a result, in cave environments, you have enriched near-infrared region. So in caves, there are some microbes, they have evolved to harvest that near-infrared region for their phototrophy. So I'm trying to understand whether they have new pigments to harvest that near-infrared region, and then whether they have specific genetic mechanisms to do that. So basically, I'm trying to understand how phototrophic life, or how these cyanobacteria or green algae have adapted or evolved to harvest that invisible region or the infrared region for their phototrophy.
So you're looking at the microbes in caves here. Is that to see if they can? There's possibly microbes that can use that light source on other planets?
Yeah.
Or?
So the thing is, so for now, for this case, I have to go into the universal scale. So in the universe, there are billions of stars. So these stars have categorized based on their surface temperature and their size, and there are so many other categories. So one of the most used categories is we categorize them based on their surface temperature. So based on that category, we say there are several groups: M-type stars, K-type stars, G-type stars, so on. So our sun is a J-type star. So our sun emits more of its lights in the visible light region. So that's why our life have evolved to harvest that specific region, because that's what we receive most on our surface, on our planet. But 88% of the stars in the universe are not J-type stars.
Oh.
So 88% of the stars in the universe are either M-type or K-type stars. So these stars emit more near-infrared light compared to visible light region. So if there's any life surrounding these stars, let's say if there's any planets around these stars, M-type or K-type stars, so they receive more near-infrared light than visible light region. So these life should have evolved to harvest near-infrared, not the visible light region.
Okay.
Because every life forms, they try to adapt to harvest what they have most in the environment. So if there's any phototrophic life in the universe, the highest probability is to find life which use an infrared light for their phototrophy, but not visible light like on our planet.
So you're doing all your research now. You're testing the cyanobacteria, you're taking samples and doing all those things. So it's you're really getting moving on your project. What would be like, what is the overall goal, like at the end of this? Like, what would you want to accomplish? Like, what is your dream?
So the end goal is to find the upper limit of the wavelength which can support phototrophic life. Okay, so for now we know visible light can support phototrophic life, and then we know there are some chlorophylls which can absorb light energy up to 720, 740, and then there are some bacterial chlorophylls, they can absorb light energy up to 900 something nanometers. So we try to see whether phototrophic life can absorb a little more beyond that region, and we can establish a limit for the upper wavelength limit for the phototrophic life. So we can say, okay, this is the maximum wavelength that phototrophic life can absorb based on our Earth microorganisms. So that's the thing, like wavelength wise, what's the upper limit? And then we think, so me and my professor, and we are so sure that we can find some new photopigments. So let's say it can be a novel chlorophyll. For now, we have chlorophyll Ds and F. If we find something new, it's going to be like a groundbreaking discovery. So we hope. So hope for the best. We hope that we can find something new, like a new chlorophyll or something, which can absorb a little bit more into the infrared region. Or it can be something else. Let's say there are accessory photopigments like carotenoids, xanthophylls. So let's say in plants, it's not only chlorophylls. They have other pigments as well to absorb the light energy. So forget about chlorophyll. Let's say we couldn't find anything new, like new chlorophyll, but still there's a chance for us to find some carotenoids or xanthophylls, like that kind of accessory pigments which can absorb a little bit in infrared as well. Or, okay, let's say we got unlucky to find new chlorophyll or like new accessory pigment, but still there are some proteins which can absorb light energy as well. In cyanobacteria, they have a special unit called phycobilisome. So it's a protein unit, and they have different proteins. We call them phycobiliproteins. So these proteins also can absorb a little bit into red and the infrared region. So there's a higher chance for us to find at least one of those or several. So our goal is to find the upper wavelength limit for phototrophic life, at the same time to find something new, some chlorophylls, accessory pigments, or some proteins which can absorb a little bit into an infrared region, which will be a groundbreaking discovery. Wow. For sure. And then if we can find the genes which are responsible to synthesize those proteins or those pigments, we can use those genes for biotechnology as well. So that's not going to be my project, but in future someone can use those genes and they can incorporate those genes into biotechnology, and maybe, let's say, there are some algae we can use for bioreactors. So now they can absorb not only the visible light region, but also into the infrared region. So that will increase the net productivity. So we can increase the harvest because now they harvest more of the solar spectrum. So they harvest more energy. So by doing that, so they can increase the harvest and the productivity and everything. So that's going to help the world as well.
Wow, this project has a lot of potential.
Yeah, so not just astronomy, but also for biotechnology as well.
It's super exciting.
Well, maybe I'm not the one who's going to do the biotechnology part, but at least we can find the pigments or the proteins and then the genes.
Yeah, I hope you do. Yeah, it's an amazing project.
Thank you. And then we also have some other use for Martian caves as well. So, yeah, so on Mars, there are some caves. So they are not caves, they are lava tubes. Mm-hmm. So Martian lava tubes are considered as one of the best places to have life. The thing is, like, if you consider the Martian surface, during the daytime, it's really hot. And then in nighttime, it goes like freezing temperatures, like super freezing. The reason is that Mars doesn't have a good atmosphere, like a thick atmosphere, so it cannot kind of balance the temperature.
Okay. So caves can. So.
Yeah, but caves, inside of the caves, it doesn't matter what's the outside temperature, they have kind of very stable temperature inside of the caves. So if there's any life on the surface of Mars, it's going to be really hard for that life to survive because of that huge temperature fluctuation. But if there's any life inside of the caves, they can survive easily because of the very stable temperatures. At the same time, if you think of the Martian surface, so Mars doesn't have the ozone layer like we have on our Earth. So if there's any UV radiation coming from sun, so these life should have evolved to either they should have evolved against UV radiation with some specific protective mechanisms, or there's no chance for them to survive at all because they receive like 100 times more UV than we receive on our Earth because they don't have that protective layer. So it's really hard for life to survive on the surface. Temperature fluctuation and then UV, and also they receive cosmic radiation because they don't have the magnetic field. And then they have meteor impacts very often. So again, it's really hard for life to survive on the surface. So that's why caves are still the best place if there's any life. So there are some scientists, they are trying to understand the chemotrophic life in Martian caves. But what if there are phototrophic life? So that's how I can involve into Martian lava tubes as well.
Okay, wow.
So not just chemotroph, also there can be a possibility for phototrophic life in the entrance part, like I do here. So by studying our life, I can predict, okay, our life can survive in these cave systems up to this level. Well, we can apply that thing to Martian caves as well. So if NASA would like to do any future missions, they will be like, oh, so there are some data which is published by Barton lab a few years ago. Okay, they have shown the possibility of this kind of life, and these are the type of things that we should look for, and they can design a machine to look for that particular thing on Martian caves. So that's another use.
How close are we to doing that?
Yeah, so like most of the photosynthetic, not the photosynthetic life, they all have carotenoids. It's really easy to detect carotenoids than chlorophylls because chlorophylls will degrade very quickly, but carotenoids can survive for a very long time. And then there's a specific technique called Raman. With Raman, you can very easily detect carotenoids, like even with just small molecule. Even if there's a single cell, let's say there's a single cell, only just one bacteria, and if this thing has carotenoids, Raman can easily detect, it gives like a big peak.
Oh, wow.
Let's say we go study our caves on Earth, and we say okay, the entrance zone, of course, we have direct sunlight, so there can be phototrophic life. And then we go a little bit inside, that's the twilight zone, there you have reflected sunlight. So it's kind of dim, but not direct sunlight. And then you go deeper, that's dark zone. And there you don't have any direct sunlight or any reflected sunlight, so it's dark for our eyes. But if you remember, I told you earlier, because of that refraction or reflection of the near-infrared light into the cave system, even though it's dark for our eyes, there's still near-infrared light. So this life can survive harvesting that near-infrared region for their survival. So if we can find any link of those near-infrared driven phototrophy and their production of carotenoids, we can propose, okay, look for these kind of carotenoids on Martian lava tubes or something like that. So that's another goal. I mean, it's not the main goal, but that's another usage.
Okay, wow. Yeah.
So for our solar system, also out of our solar system, and also for biotechnology.
Okay, there's a lot of uses for it. Really exciting research.
Thank you.
Yeah. And sir, thank you so much for letting me interview you. And it was a really interesting interview, and I've learned so much. I can't wait to share this with other people.
And thank you for inviting me for your podcast, and I hope you guys will understand. If you guys have any questions, they can give my email to KLab, and they can email me if they have any questions.